So when you think DNA modification, what do you think? You think designer babies, cure for cancer, babies that glow in the dark even. Etc. The truth is, just about anything is possible with DNA modification, because you're modifying the literal instructions that the organism cells use to make the organism. But that's only theoretically, because currently there is no cheap, efficient, or indeed effective method of editing DNA. Not until 10 years ago, that is, when scientists discovered a protein they appropriately nicknamed genetic scissors. But I feel that the scientific name is just a bit CRISPR. Today, I'll talk a bit about what CRISPR does, what it enables, and its massive potential for New Zealand's primary sector. So CRISPR-Cas9, the full name, beyond its capacity for terrible puns, is, like I mentioned, a scissor-like protein. The process of actually editing DNA involves, firstly, cleaving the DNA, and then inserting the desired section of new DNA. And why is CRISPR-Cas9 so revolutionary? Well, because that's exactly what it exists for. It's found as the immune system of the bacteria Streptococcus pyogenes, where it cleaves invading DNA deemed harmful at the exact spot it deems harmful on the whole DNA strand. You can change what CRISPR thinks is harmful, and thus cleave any DNA. And this solves the two biggest setbacks with the previous DNA cleaving method, called a restriction enzyme. Firstly, REs only cut at a particular DNA section. So DNA is a sequence of unique code, and different REs cut at different sequences. If you can't find the RE that cuts at the sequence you want, you're out of luck. Furthermore, because they cleave by sequence and not by location like CRISPR, they cut the DNA every time the sequence appears, most likely killing the cell because you've cut it too many times. It's like running a piece of paper through a shredder because you want to cut it in half. Thankfully, with the discovery of CRISPR, we now have scissors to cut the paper. So what does this mean for DNA modification? It means that now edits we make to a sperm or egg cell can actually function in the resulting living organism. Genes and traits can be customized and exchanged between organisms. And the most exciting application of this to New Zealand is without question our primary sector. So how can we apply it to so large a scale? Well, this will sound quite familiar to you. About 1 hour, you heard this about 1 hour ago from Isaac. Over time, genetics has naturally perfected a trait inheritance system called a gene drive. So most genes, now normally most genes have about a 50% chance of being inherited, 50% chance of genes from the mum being inherited, 50% chance of genes from the father being inherited. But some genes have evolved mechanisms that allow them to be retained nearly 100% of the time. And these are called gene drives. We can mimic these mechanisms to manually retain CRISPR-modified genes of our choosing, release the modified organism into its habitat, and very quickly spread the gene through its population. With gene drives, we can now solve leading issues with agriculture in New Zealand, such as invasive species, pollution of water and soil, and even crop enhancement. And I'll pique your interest about a few of those today. So first off, invasive species. They damage and feed on our native flora, they prey on certain species of native fauna, and most importantly, they spread harmful infections to our cattle and sheep, which make up a substantial portion of New Zealand's economy, making them hugely detrimental to our agriculture. An example of a significant pest is a name you'll have heard before, the pictured common brush-tailed possum, which transmits the tuberculosis-causing M. bovis bacteria vigorously to sheep and cattle, and has been responsible for more farm animal losses than all other diseases combined. Thankfully, CRISPR gene drive methods that are already being practiced on mosquitoes can similarly be applied to the possum or any other of our pests. In 2015, researchers from the University of California successfully conferred a gene drive to a test group of mosquitoes that made them resistant to malaria, meaning these mosquitoes are now unable to contract the disease and thus unable to transmit it. Or another strategy developed in Imperial College London introduces a gene drive that causes sterility in female mosquitoes, equally effectively controlling the population of mosquitoes as opposed to the disease. So by utilizing strategies on New Zealand's invasive species, the government's goal of being predator-free by 2050, set July last year, is now very much achievable. Next, pollution. An increase in intensive dairy production and extensive use of agrochemicals like fertilizers have resulted in greater levels of inorganic nitrogen, which is the definitive measure of pollution in our soil and rivers. Water pollution is not only damaging to surrounding ecosystems, but to the health of its consumers, aka us. And soil pollution is damaging to crop success as well as pasture for livestock, both of which have a huge impact on our agricultural output. But you guessed it, CRISPR-Cas9 gene modification can be used to eliminate inorganic nitrogen pollution. And the solution is a perfect showcase of CRISPR's versatility and flexibility. Azotobacter, a form of bacteria found commonly in freshwater, in neutral soils and freshwater, is capable of fixing nitrogen, or in other words, converting it into ammonia. The most common form, however, doesn't achieve this efficiently enough to be of substantial benefit. Thankfully, though, there exists a mutated variant of Azotobacter that can fix nitrogen at temperatures as low as 5 degrees Celsius and is 10 times more active. So not only can it fix nitrogen far more efficiently, it can also function in water. So what can we do with this? Well, you know the drill. Enforce a CRISPR gene drive of the variant into a sample of Azotobacter, which can then be applied and spread into contaminated sites. Applied to soil, the ammonia produced from the nitrogen acts as a natural fertilizer, which promotes leaf growth in affected plants. And in water, ammonia and chlorine make chloramine, which can purify the water. And because we now have means to access any region of DNA with CRISPR, we can kill both the pollution bird and the crop enhancement bird with one CRISPR stone. So, it goes without saying that everything I've mentioned is merely scratching the surface of what CRISPR-Cas9 has made us capable of. Generating a better understanding of genetic code will be the next step in genetic modification, after which the only limit to what CRISPR-Cas9 can help us achieve is our imagination. And thus we can drive New Zealand to becoming the most beautiful, stimulating, exciting, or perhaps even glow-in-the-dark place in which to live. Thank you very much.
Thank you, Joshua. Isn't that great to have the final speaker quotes Callaghan, who was the motivation for this whole competition?